So my name is Marco Brandizi. Just to give you a few information about my background while I go full screen with this here. I'm basically a software engineer. I've been working in essentially with server-side data manipulation and software for doing that in the biomedical field. And as such, I'm interested in providing in open data and linked data in particular in this field. But I'd like to start with putting it into context, and to me, a general context where we are is the one of openness that has spread over the last 10 or 20 years or so. And it is about many things as you see in these slides. We started with software and other intellectual and immaterial productions like publications or knowledge. And recently we have had other forms of openness in concrete things. This is a picture of Arduino, which is a popular open electronic device to build embedded systems. So we have consumer products like Open Cola, which is essentially like Coca-Cola, but with free and open recipe. And even The Economist is mentioning businesses like Airbnb or Uber. That is a bit about that. Now, today I'm going to talk about open data, which again is more general than the biomedical field itself. For instance, many governments have started for a few years now projects to provide structured, machine-readable data mainly about public administration. Our field, as probably most of the audience knows, has been doing that for years, and I think we are more advanced than in other fields. And that's because we have strong needs to share data and to share them in proper forms, because we have heterogeneous data to study, to integrate them, and so forth. And in fact, you can see several examples of that kind of sharing. Here at EBI, we provide several repositories for public experimental data in life science. We already used advanced approaches like ontologies and linked data, which is what I'm going to mainly talk about. Now, open data can be provided in various forms and with different degrees of machine readability and sophistication. It ranges from almost nothing, like the PDF, to CSV files. What I'd like to talk about today, as I was saying, is the most advanced forms of open data, which are based on the approach of linked data and on the specific technologies of the semantic web. The idea about that, I don't want to do training on this technicalities and the specific topic. Also because I have seen the audience is at least have a minimum familiarity with that. But anyway, just to make a little summary, the idea about linked data and semantic web in the specifics is to reuse the traditional web that was designed for documents, which are supposed to be directly consumed by users, to reuse it to publish structured and machine-readable data. And also to include meaning and semantics in such structured data. So this is a very quick summary of how this is realized by means of the technologies which goes under the name of the Semantic Web, proposed by people under the W3C consortium, some of the same guys behind the traditional web. And I'm just summarizing the basic ingredients of this technology, which goes under the name of the RDF, essentially the resource description framework. So one ingredient is to use universal identifiers, and one way to reuse the traditional web is to use URIs, which are simply put are variants or traditional web URLs. URLs can identify web documents. URIs can identify basically anything, real entities, concepts, and so forth. Then we can use this basic building block of the statement or triple or binary relations. There are different ways to see it, to relate to URIs, that is the things or call it resources in the field, to relate to different resources by a particular relation. So this is a way to extend the traditional hyperlink concept with some sort of typed link that say why the two resources are related one each other, while this was not possible with traditional web documents. You only had a generic link for them. And similarly, you can do that for scalar values, strings, and numbers. And you can add ontologies on this, that is, provide meaningful semantic descriptions, so the description of the meaning associated to existing resources, by using the same mechanism. And probably you are really familiar with this part of it. Moreover, you reuse the web by just providing resolvable URIs, and the dashed boxes here should give an example of that. So you can imagine that, for instance, inside the blue box, you can have that the URI about this protein is providing statements describing the protein itself. And when you use a client to fetch such statements, similar to the way you fetch a traditional web document, you get exactly them encoded in a proper form, that is RDF encoded in a proper form like XML or other languages such as SPARQL. And the fact we use universal URIs allow us to, for instance, to fetch graph fragments from different servers, that is across the web, and to put them together almost automatically in practice, and to build arbitrarily complex graphs like in this figure. So this is a very quick summary of what the technology is. There is much material to get a deeper knowledge about that. Some of them produced by myself and my organization as well, and I refer you to these links I reported. Well, what can we do with this, in addition to represent the reality as much as possible without little distortions, as little distortions as possible, as this figure tries to hint? Well, I can show you what is possible to do with biomedical data, taking the example of the European Bioinformatics Institute about that. So we basically have the need to represent life data, life science data. And as EBI, we are organized as different groups and different data repositories. Each works quite independently, and we have connections about relevant relationships and relevant data integration. So what we have done about 2 years ago now in the ABI RDF platform group is essentially to mirror this kind of organization. And for the nodes you see here that actually represent such repositories for each of these nodes, we have built a so-called triple store. That is a data set made of RDF triples or statements, as I've shown before, by means of connections. And here we are representing the main of such connections. Personally, I work on the BioSamples database data sets, which I'm going to talk a bit more later on. In doing this, we have tried to use existing ontologies as much as possible and to try to use them in a coherent way and to obtain data integration among other means by means of ontologies. And this is a schema that gives you a portrait of the kind of integration that we have between the different data sets, and also with other data sets external to the EBI itself, some in the Elixir project, some other in Bioinformatics, and so on. So what can we do with this? In addition to exploring your resolvable URIs, we can use a standard language that has been designed to query this kind of data, and which, as maybe you already know, is named SPARQL. And so here I can show you one of the most complicated examples that you can solve by means of this query language. I would not recommend to start studying the stuff of this data by a query as complex like this. Here I'm just giving the example because it allows me to show you the power of this approach and an advanced example. So as you see, we have a drug. We want to match relevant targets for these drugs, and we want information about gene expression and pathways about that. So we are involving different data sets, and the way that SPARQL is usually served is providing a web service for each of these data sets to, and you can send SPARQL queries using SPARQL language to one of these web services, or as I'm going to show, to multiple SPARQL web services, also named endpoints at the same time. So this is a glimpse of how the language works. Again, I don't want to do a semantic web course here, but very briefly, for instance, if you see this assay targets, these two-column names are just shortcuts for longer URIs. You see that is essentially a graph that has the purpose to match, a language that has the purpose to match graphs. So it's essentially a graph matching language. And so here triples in this form having a fixed statements will be matched. And if you combine this with the next line, you will see that graphs having target in the middle will be matched as well. This is the first fragment of a rather complicated query, and this fragment will be executed against the Campbell endpoint to get information about drugs and proteins. Then we are linking other data sets, for instance, by reusing URIs about proteins coming from UniProt, and these other fragments will match graphs from the triple store in the Reactome data set about biological pathways. And so the syntax is the same. The addition is this service keywords, which can be used to just specify an additional endpoint. And so this mechanism, when it works, because it's still a bit problematic from the technical point of view anyway, in this case it works. It usually works at least. And this mechanism is rather powerful and flexible and can be flexibly used to query multiple data sets with a uniform language. And this is the last fragment that additionally queries information about gene expression in the Gene Expression Atlas endpoint. So the overall thing is like this. Let's see if okay, I don't know if I have enough time, and again, I can refer you to this link if you want to try print yourself on the live thing. So as I was saying, I personally work on the BioSamples Database project, which is first of all data collection supported by Oracle, and a website that users can come to and browse different type of information that is focused on biological samples. Essentially, we import data from existing repositories which are assay or technology specific, as you can see, ENA or TRIDE or ReExpress are examples. We also import other biosample from other biosample repositories or from repositories of cell lines and so forth. And the idea is that you can use this resource to perform sample-based queries and to possibly navigate to other type of data. So we, as in other cases, we converted this into an RDF type of store and set up an endpoint. And this is another example of how this technology can be useful. This is a work made in the last biohackathon in Japan. They have developed this JavaScript library, D3 SPARQL, which is in turn is based on the D3 library, and it is essentially to perform data visualizations. And here you can simply send a SPARQL query from Japan, I guess, to the EBI and get information about samples which are geolocated. We have collection coordinates for a number of them, and eventually you can use the library to show these results by just using the standard. So they didn't ask much support from ourselves here. So just to give you an example of how much this can be flexible. Now, the next thing I'd like to show is a question I'm been investigating, and is at which point we are with this. How good the modeling by means of these approaches is good, how useful are these data, and how easy it is to query them. Now, I guess you understand this is a very wide topic, so I'm only going to consider a few aspects like modeling, linking, and navigability and usage of URIs and availability of applications. And I'm going to consider this for just the API data, for a sum of the API data and similar or related data. So for instance, on the point of modeling, if we consider the way samples and assays as well are modeled, we see, first of all, that a common approach is to use is to define custom schemas typically for each data set, and to reuse ontologies by extending existing ones. And this is not exactly fully reuse of existing ontologies, but at least is something in between. And it's some intermediate solution where you have this indirect reuse of existing ontologies. And the next thing about this is that this is one of the two approaches I've seen around for modeling samples and real samples compared to other visions that have people like the OB developers, for instance. And this approach consists essentially in describing the samples in terms of pairs of value and type for each of the features the sample have. So here, for instance, you have that the organism is described with a text value that is indirectly in this case, but anyway, is attached to an existing ontology term, ontology concept about the type of the feature that we are trying to represent, so the type of organism. And if you take the example of the gene expression atlas data, the approach is rather similar, a bit simpler, and in fact we are planning to adopt the same, to simplify the same way BioSD as well. But anyway, the approach is rather similar, but yet the schema is not exactly the same. So interoperability is still possible, but it's not exactly like the ideal, like the vision that Semantic Web proposes endorse. And so similar considerations can be made for other data sets that are around, and here there are a few links about that. The situation is even more heterogeneous than the two examples have shown, because, for instance, in this case, coming from the master clinic, the type of property and the type of feature that is attached to the sample is modeled by means of the property instead of using ontology concepts or value type pairs as I've shown before. So this usually creates problems. For instance, Frank van Armelen, which is a semantic web expert in the field, has once put it such by saying that these technologies do not solve the interoperability problem, they more put it on the table. So we can possibly do more. There is maybe more research to do in this direction. This is another example about gene expression taken from a rather known paper, where both samples and assays and even gene expression data are modeled differently, and you have a possible solution to uniform these different representations, and so that you can use uniform queries, which is basically based on query rewriting rules. So things go a bit better on the point of URIs and the ways they are defined and reused. We even at EBI we have a common policy for them. We have a fair degree of URI reuse. As I've shown before, we have a fair use of common ontologies. So they are the same ontologies are reused across different data sets. And we have adopted common approaches like URI resolutions by using the same syntaxes and so forth. Part of this is supported by the same technical infrastructure that we use for each data sets, and in particular, we use this navigation application that is named Lodestar, that basically is something you can put on top of an existing SPARQL endpoint, and then it can provide you a nice web interface to perform SPARQL queries if you are a human being, or it can do services like URI resolutions either by means of techniques like content negotiation. So this brings me to the last point of the applications. So this screenshots here is from this Lodestar interface, and if we look at our data and at similar data like OpenFacts or Bio2RDF, the kind of application that is most common is the one that is dedicated to the technically wise user. And maybe we are missing applications that are focused more on the biologists. OpenFacts, I think, is one of the relevant exceptions. We have something in between, like this. I think is named SPARQL graph, where you basically can build queries in a visual way, which is a bit a step ahead. The mention of D3 SPARQL is another exception, although it's still dedicated to the developers. From here, I have a few suggestions of new directions we could take with the existing RDF data at EBI, and I really would like. I'm really open to possible collaborations. In particular, the ones that could be possible around the BioSD data set. And so just to mention a few examples, in addition to the ones listed, so one thing is searching biological samples by means of feature similarity. This is a very simple case that we tried with a few students, where basically we count the number of similar features up to a certain count. But of course, more can be done. And more in general, another interesting thing that we haven't done so much so far with EBI data is to take a concrete biomedical cases, biological cases, and to use the data to answer relevant biological questions. So this is an example from the HCLS group, which is quite known in the field, and they basically build the data in order to answer a question that involves different biological entities and different existing data to get to biologically relevant results. And so this is something that would be nice to do with EBI data. Then finally, there would be other considerations to make about ontologies, which have been talked years ago around description logics and around the now language, which is very advanced compared to what we are actually using today. And probably things are going to remain this way, because personally I think OWL is really too complicated. Well, no, it's really, it has technical and conceptual problems to be easy to apply it on the real web, even the semantic web. So it's still good that we can use certain features about automatic reasoning and so on. So almost finished. I'd like to thank a number of people. The RDF platform at EBI has been built by a group involving different people from different specific repositories. So Andy Jensen and Simon Jupp and James Malone are the ones I've collaborated mostly. In the BioSamples team, I'm thankful to people like Adam Falkenbridge, Drashti, Tony Bartlett, Mike Gostev, and Helen Parkinson, and my team leader is Ugis Sarkans, which coordinates the technical work in these projects. And our funder, as probably you already know, is the European project Bioinformatics. And finally, thanks to you all. And as I said, I would be interested in possible collaborations. So now I'm going to read a few questions here. So one question by Paul is about the performance with SPARQL. Well, this depends. First of all, compared to a few years ago, generally speaking, things have quite improved. And to give a figure, we can perform queries in seconds over even billion triples. which is comparable to an average, a medium to big size traditional database. The thing is, in my experience, is still quite variable. It's quite depends on the query, and also you can have technical problems like the server load, and Virtuoso. We have seen that Virtuoso sometimes, at least in previous versions, has issues like memory leaks when it runs for a while. So I would say overall the performance is acceptable for medium needs. There is still to be things are still to be improved. And another thing comes in my mind is that a few people, for instance, from the chemistry community, I remember Colin Batchelor, are even questioning this idea of making SPARQL endpoints public. And for example, the OpenFacts projects has chosen not to do that. So they still have SPARQL and they use it internally. They allow access to their data by means of APIs and basically CANet queries and things like that, which is a bit less flexible on the one hand. On the other hand, it avoids certain problems, namely performance problems. Okay, so as last thing, I can show you the live part that I skipped before. Let me see if I can go quickly back. So this is the Lodestar interface. It's quite generic. You can download it and set up with a number of things. First of all, the SPARQL endpoint against which these queries are sent, and then layout stuff, examples here, and so forth. So this is the query I've shown before. As I said, it goes through different data sets. I hope it's still working. I've seen some problems in the Tom computer. Okay, should still be working. So when the query is performed against the SPARQL endpoint, the interface shows the results in this form. For the human beings, of course, if you send this same URL to from a client, the interface behind automatically recognizes that, and it provides results in this other form, which is essentially XML and is suited for your client applications. So let's go back to this. So from here, you can exploit another feature of Lodestar, which is its ability to show a human-readable form of URI resolution. So basically, these are statements about this URI, which is about these proteins, and it shows you these statements of which the URI is subject, is the starting point. And again, you can use a technique that goes under the name of content negotiation. So if you use a client and you specify you want this turtle format in the header, you have something that is actually machine readable. So I have next questions. What dictionaries are we using for ontologies? For the ontologies we use, there is the schema that I've shown before, and also if you go data set, one data set at a time, in the documentation page, they usually list at least the main ontologies that we use, like in this example. And every page also show the custom schema that was defined for the specific data set, plus, as I was saying, the ontology being used. And these ontologies are basically the ones available for the entire world. Some of them were developed here at EBI, like EFO or KEBI, many others were developed around the world and published, and a good resource to browse and search for ontologies is our ontology lookup service and also bioportal.